It's amazing to see all the great things that are going on at DARPA and to share them with you. And I'm going to tell you today how an organic chemist came to embrace biological complexity. But at first, I'm going to give a caveat, okay? My formal training in biology ended in the 9th grade. So the musings that you're going to hear are from a high school dropout, so take that for what you will. And I'll tell you why it ended in the 9th grade. Because when I took biology in high school, it was primarily a descriptive discipline. It was at the organismal level, right? You named stuff. You may have studied stuff by cutting it open. So I cut open worms and sheep's eyes and thought that was disgusting, thought that was the entirety of biology, and said, JPG, out, done, okay? So then I said, all right, well, what do I want to do? I knew I wanted to be a scientist. So I flirted with physics for a while, thought about maybe being a particle physicist, you know, this idea that I can understand everything. If I just understand what's going on at the level of quarks and gluons, I would somehow have some insight into how the world worked. I was a pragmatic person, though, and I realized that probably wasn't going to happen. And at this point, this was before physicists went to go work on Wall Street and make zillions of dollars. So I took a middle road, okay? I said, I'm going to be a scientist, not going to be a physicist, not going to be a biologist. I decided to become a chemist. Okay? And I liked chemistry for a lot of reasons. I mean, you like burning things and blowing stuff up. But ultimately, what I really liked about it as a fundamental level is that it is a synthetic discipline. Okay? It embraces not just analysis, so studying the nature of something, but synthesis, using it to produce something useful. And I really, really liked that. So I knew I wanted to be a chemist. Studied it in high school, studied it in college. In college, I made a lot of materials, and when I went to graduate school at Caltech, I knew I was going to use synthetic chemistry to produce useful materials. And I knew who I was going to do my PhD studies with, a gentleman by the name of Dennis Doherty, who at the time had been making magnetic materials, not out of hard things like iron and so on, but out of soft things, organic, carbon materials. So I would go to Dennis, you know, first-year graduate student, all cocky, say, Dennis, I want to use chemistry to make materials. And Dennis says, not so fast, hombre. We're not doing that anymore. In fact, you know, we're winding that down. We're studying a new problem. We're going to study the most complex device on the face of the earth. We're going to study the brain. Okay? And we're going to do this at the molecular level, okay? By using chemistry to understand the ion channels and the transporters, we're going to understand things like thought, you know, feeling, consciousness, all these big picture things. This is the kind of stuff that professors sell to first-year graduate students, and people like me buy up. Now, mind you, this is a time in my life when DNA in a seminar title meant do not attend, okay? I had a 9th grade education in biology, but I signed on, okay? So we're going to study the brain at the molecular level. How are we going to do that? Are we going to do it in the brain? Unfortunately not. We are going to do it using these things. Some of you in the audience probably know what these are. These are Xenopus oocytes. They are the immature egg cells of the African clawed toad from the species Xenopus. And what they are useful for, in addition to making more African clawed toads, is that they are physically large cells, okay? And it turns out that you can inject nucleic acid, so either DNA or RNA, and these cells will obligingly translate that genetic material into mechanical things, the machines, the proteins of thought and emotion, ion channels and transporters. Moreover, you can study these by sticking 2 electrodes into these cells, applying a voltage, applying a drug, and measuring the changes in potential as you apply that drug. So I did this for a large part of my PhD, and I concluded several things at the end. The first is that after injecting probably hundreds of thousands of Xenopus oocytes and sticking them with 2 electrodes, I never wanted to do that again, okay? And I can assure you I have not since. I wouldn't recommend it to any of you either, but you know, maybe some of you are into that sort of stuff. I don't know. But the second thing was that this ultimate reductionist view of trying to understand neuroscience probably wasn't going to get us to where we wanted to be. It's not to discount it. It's important and it can solve a lot of problems. But the understanding of consciousness is probably not going to come at this reductionist level of ion channels and transports, even though they're important, much in the way that the nucleus is important is there in chemistry, but is not so critical to understanding organic chemistry or reactivity. So we have to worry about these levels of abstraction at which we solve problems. Okay? Then sort of the biggest lesson I learned is that biology is really useful. These cells obligingly did my bidding for me. Okay? They could produce interesting things, and I really liked that. So when I went forward in my career, in my postdoc, I started thinking about more hardcore chemistry again. How do we produce molecules? And I would sit around and talk to the fellow postdocs about how there's an entire discipline of synthetic chemistry, people who dedicate their lives to making things that exist. This is a molecule known as an apotholone. It's a potential anti-cancer treatment. And to the chemists, they find this appealing because it's difficult to make. Okay? Now, from my perspective, I look at this and say, yeah, it's difficult to make, but it's a natural product. It came from an organism somewhere that's probably been dutifully churning this out for millions of years without expending a whole lot of effort. Okay? It didn't take, while it might take a six-pack of postdocs in the laboratory to produce this, single-celled organisms already knew how to do this. And as somebody contemplating an academic career and realizing that bacteria are inexpensive and postdocs are very expensive, I wanted to take advantage of how nature did chemistry. So how can we harness the power of biological systems? And in this case, it actually is a relatively straightforward reductionist problem. We look at the molecules, we ask how it's made. Ultimately, they're encoded by genes. We can isolate those genes, we can sequence them, we can stitch them together in new ways, stick them in other organisms like E. coli, and at the end of the day, we assemble the pathway, we stick it in the organism, and it produces the product. Okay? So we can use biology as a synthetic discipline. So I became a synthetic biologist. And in my academic career, I spent a number of years, over a decade in fact, trying to figure out how I could control cells to do my bidding. So turning genes on and off using small molecules. If you want to talk the details later, we certainly can do that. But essentially, we were using small molecules interacting with RNA to turn genes on and off. And after a long time of doing this, so engineering bacteria to do stuff, make stuff, detect stuff, whatever, I got worried. Okay? And I got seriously concerned about the discipline of synthetic biology. After going to many, many meetings, I realized that the systems that we were building ourselves, and indeed everybody else's, were very, very fragile. Okay? So if we're to operate, if biology is to be an engineering technology that makes it beyond the lab, beyond the petri dish, we needed to build systems that are substantially more robust. And the reason that these systems, I think, were relatively fragile is A, they were human design, and they were sort of the first generation of them, but also we separated the real biology from the engineered biology. Okay? So we start with organisms that, while once evolved in the wild, hadn't seen the inside of a mouse in 50 years. We took laboratory strains of E. coli because they're easy to engineer. But as a result, they were relatively weak. So this separating the real biology from the engineered biology is a problem. We do this, of course, everybody does this. So if you're trying to engineer an organism, you introduce some genes, you need to coddle that organism. So you put it in a nice flask, you give it rich medium, you add antibiotics to keep competitors away. In this case, you give it a light to keep it happy, and so on. So this is maintaining a physical separation between the engineered biology and the rest of the world. And of course, there are reasons to do this, but in this case, we did it simply because the organisms were so weak they wouldn't survive. So the predominant thinking in this area could really be summed up by this quote from Robert Frost, which is, good fences make good neighbors. Okay, so this is over 100 years ago. Separating the engineered from the natural biology. And that works in several contexts. Okay? If we're studying ion channels and oocytes, that works. If we're using E. coli or yeast to produce compounds, that works. Maybe not quite as well, but it works. But we want to go beyond that. So when I came to DARPA, I wanted to first deal with this project of the fragility of biological systems. And second of all, this idea of just using the organisms as a chassis to do something and then tossing the organism into a bucket of bleach at the end of the day seemed to be leaving a lot on the table. Okay? We were just using the organism as being complicit with what we're doing, but not taking advantage of all that biology has to offer. Okay? So the organism shouldn't be along for the ride. We should be, instead of isolating biological complexity, we should be embracing it. And that's what I'm doing at DARPA. So we recently launched a program. It was in response to this idea that the world is a complex place. It's more complex than a petri dish. Whether it's the desert, the sea, the forest, or inside your gut, it's not a sterile environment. And if we want to build systems that operate in any of these environments, we're going to have to do a hell of a lot better than what we've been doing. So we launched a program called BRICS, Biological Robustness in Complex Settings. We're in the source selection phase of that right now, but essentially the goal is in microbial systems to build not just single organisms, but microbial communities that can operate safely and reliably in fluctuating environments, okay? Why do we want to do this? Well, unless you've been living under a rock for the past several years, you've heard a lot about the microbiome. Rob Knight, one of the experts in this field, is sitting right there. And we've come to realize over the past several years that the organisms that grow on us and in us affect us in profound ways that we don't completely understand. But if we want to ever think about manipulating them, whether it's to impact our immunity, how we digest our food, how we think, we're going to need to solve these problems of engineering microbial communities. We're not limited to working just inside the body. One of the things that I'm very excited about is this notion of solving problems in agriculture, so in plants, but doing it at the level of microbes. Okay? So a lot of people, for better or for worse, are uncomfortable with the idea of genetic engineering of something that grows above ground, something that you would eat. But as many of you know, and as we heard earlier, symbiosis is very important in biology. What helps plants grow is often what's in the soil. So the microbes that are there that are taking cheap, free nitrogen from the atmosphere, fixing it, and helping these plants grow. So are there ways that we can engineer robust microbial communities that help increase and support plant growth? This is good for a lot of reasons. It avoids GMOs for foodstuffs. And fertilizers are very, very expensive, okay? So it turns out that about 3 to 5% of the world's natural gas supply each year goes to produce fertilizer. That's 1% of the world's energy supply. If we can make even a dent in this problem, this is huge, okay? And I think we can start doing this and using engineered microbes to help. Fertilizers have other problems. I'll talk about that in just a minute. So, of course, you know, why do we want to engineer these plants as well? Why is DOD interested in this area? Food security is a huge problem worldwide. About over 800 million people each night go to bed hungry. Okay? And to feed 9 billion people on this planet by the year 2050, it's estimated we're going to have to increase agricultural production by 60%. And if you look at the areas that suffer from food insecurity, a lot of them are in areas that the people just don't like us very much or are unstable for other reasons. And that's a threat multiplier. If people start fighting over food and fighting over water, that's problematic, and the DOD is going to have to come in and help with that. So if we can address that problem early using science, I'm all for it. Water security is similar. If you were to look at water insecurity across the globe, you would see a similar map to the one I showed you for food security. This is actually a different map. That's a map of northern Ohio taken in 2011. And in the lower left, you'll see an algal bloom. It's actually cyanobacteria near the town of Toledo. So in the year 2011, in the United States, a town of 500,000 people or surrounding regions went without water for 3 days. Not just drinking water, you couldn't even bathe in this. And why did it happen? This is self-inflicted. The reason that this happened is global warming, so water temperatures are higher, support the growth of these organisms, and fertilizer runoff, okay? If you put all these nutrients in the ground chemically, they eventually get into the water where they feed other things that grow quickly and cause a lot of problems. So if we can start using engineered microbial communities to address the fertilizer problem, we can also impact water security as well. All right, so what are some of the things that I'm thinking about going forward beyond microbial synthetic biology? I'm just going to share with you 2 areas. The first is harnessing and sensing biological systems for sensing and actuation, and then the second is building with biology. So the agency that I work for is known for many things. We heard about them this morning, but one of them, of course, is robots. Okay? DARPA is very good at building robots. Here's one that was supported. This is the Robo-Bee. And although this is a relatively simple robot, it can fly, which is more than I can do. Okay? It doesn't fly very well, but it can fly. But if you start thinking about the properties that we might want this robot to be able to do in addition to controlled flight, there are a lot of them. Okay? And in fact, they're inspired by things that already know how to do these things. So this can fly too, and it can fly very well. And it can do other things, like sense near single molecule concentrations of small molecules in complex environments. If you've drank a glass of wine outside in the summer, you know this, because the fruit fly will always find its way into the wine. In addition, they can see optical sensing, learning and memory, sophisticated actuation. So rather than try and build a fly from the ground up, why don't we embrace the complexity of this fly, take advantage of it, and then augment it? Now, this is a problem for synthetic biology because this is a much more complicated chassis. It takes a longer time to grow. It's not as obliging as some of these single-celled organisms. Ultimately, this fly has to go about its fly business as well. So as we start dealing with it, we need to make sure that a fly is still a fly. But I think there are lots of challenges and opportunities going forward for engineering multicellular organisms. The second area is building with biology. Living systems have a lot of great properties. They're ultimately solar powered, they exist at ambient temperature, they can sense and respond to their environment, and they can heal themselves. Now, the notion of building with biology is not new. We've been doing this for a long time. But the problem is that most of what we build is inert. So we've taken this remarkable living system that can do all those great things I just told you, and we strip them away. We take the tree, which is living, and then we cut it down so it's dead, and then we kiln dry it so it's hard and it doesn't do much. Okay? And as somebody that owns a wood-framed house with wood siding, you know that it would be really nice if you had siding that healed itself. Okay? So how can we start building with biological materials where instead of rendering them inert, so getting rid of all the good stuff, we leverage that to use the functional properties of biology in our designs? There are companies out there that are starting to do this, whether they're building bricks, building materials, starting with sand and with microbes, or building packing material from mycelia, from mushrooms. This is great. We're at the very beginning of this, but these are not engineered materials, and moreover, they're still inert, in some cases for defensible reasons. But if we start adding on a layer of complexity, the ability to engineer new properties into these materials, okay? So for example, in producing bricks, what do we do today? We kiln dry things at very, very high temperatures, right? Building materials are expensive to make, whether it's concrete, whether it's steel, whether it's bricks, high temperatures. Can we do this at low temperature using engineered biology? I think we can, and we can start impacting the physical world. Reduce costs, add function. So I'll just close with this idea of where I'd like to go. I'd like to go towards this idea of having essentially a blank slate for biology. How can we program living systems using biology? And what I want to suggest to you is that we're able to engineer today E. coli or Drosophila or corn. But the products still look very much like the starting materials. We're not building these from scratch yet. Okay? But if we start thinking about, instead of building on these natural systems, using them as chassis, can we go for a chassis-free synthetic biology? Much in the way that stem cells ultimately develop into complex organisms, can we start programming biological systems de novo to produce biological materials with outstanding properties? So what this is going to mean is that we're going to have to go up a level in complexity. So genes are going to be important. This reductionist is certainly important. But we need to engage new communities, and that's what I'm hoping to do today. We need people that understand developmental biology. We can't just be the gene jockeys that work in E. coli and yeast. We need to understand how multicellular organisms work together. And I'm asking for your help in this area. We want to embrace biological complexity. So finally, how many of you are familiar? You've seen this quote before? It's probably the most memorable quote from the poem, "Good fences make good neighbors." But the point of the poem from Frost is actually probably best rendered by its first sentence. Something there is that doesn't love a wall, that wants it down. The whole point of his, so if you read this, and I would encourage you, it's free on the internet, Mending Wall. He's walking around, and they go through this routine of repairing the fences every single year. And it's not clear why these fences should exist in the first place. They don't have cows that are traversing and so on. So the question is, why do we have these walls in the first place? So what I'm asking you to do is to help me break down these walls in synthetic biology as we go forward. Thank you.